Hi, welcome back to yet another revision stream. This is a biology one, and what I want to go over today is something which has been bugging me for a while. I want to go over everything to do with plants, so things like transport of water, transport of sugars. Then we'll also go on to gas exchange, things like the guard cells, literally everything that we've covered in terms of plans, I want to go over in this stream. Because even things like the structure of the xylem and the phloem, I forget. I completely forget what they look like and what they do. So I have this textbook, and to be honest, I don't like this textbook at all. This sucks. So I'm gonna go find a better textbook quickly. Because I know how transpiration and translocation works, but I wouldn't know how to describe the structure of the xylem and phloem, aside from the generic stuff, like, water does it adhere to the xylem walls? And there's companion cells in the phloem. That's all I would remember, to be honest. That's it. So let's go into classwork and let me quickly go find transpiration and cohesion. Let's look at this. So this does link to gas exchange. I don't know how prevalent this was in last year's paper. To be completely honest, when I remember doing paper 1, which was the A-level paper 1 from last year, we did it as a mock in December. I don't remember there being any questions on transpiration and translocation. And I don't remember it appearing on the AS paper. So, I feel like it could appear. Anyways, how many properties of water do you recall? This is the starting question. Obviously, you have oxygen, hydrogen, hydrogen. Now, from what I remember, isn't hydrogen positive, oxygen negatively charged? I seem to recall that. There are weak interactions. Oh my god. There are weak interactions between the hydrogen bonds, which allows them to be cohesive. So due to the interactions between the hydrogen bonds, one property is that they are cohesive. Other properties include having a large latent heat capacity, basically enabling them to act as a buffer because a large amount of energy is required to raise the temperature of water. What are some other properties? Obviously, it's important in hydrolysis and condensation. In other words, it is a metabolite. Other properties. Cohesion. So what I'm thinking here is also about the essay question. I'm thinking about the essay question here. What if we got asked in the essay question, the importance of water in biology? I'm thinking about points here. Cohesive, you could turn cohesive property, you could talk about transpiration. And the importance of that would be that it allows water to go from the roots to these photosynthesizing cells in the leaves. Large heat capacity, you could link that to enzymes, the idea that plant enzymes, or just enzymes in general, need to function at their optimum temperature. Therefore, it acts as a buffer solution. Therefore, a large amount of energy is required to raise it, meaning that enzymes aren't going to be denatured. Wouldn't really know what else to talk about there, but I don't know. Then finally, hydrolysis and condensation, you could talk about how that, joins monomers together to create polymers. For example, in DNA replication, where condensation is obviously used to form the phosphodiester bonds, or the hydrogen bonds between DNA, which is obviously joining complementary bonds. What's another property of water? Let's have a look. They are cohesive, which helps them travel through the xylem. They are metabolite. They're solvents, so ions can dissolve in them. That's an important property, but I wouldn't know how to yap about that for a whole paragraph. You could mention just its importance generally as acting as a, solvent. Most metabolic reactions happen in this solvent, such as the cytoplasm. But that's basically all the properties of water. So obviously plants need to maintain structural rigidity. That's an important property. Is in plants, the plant cell wall needs to remain turgid instead of flaccid, because then it's rigid. So if it has a high turgid pressure, this is going to be a rigid plant structure, rigid plant cell. Cell wall, and therefore it is going to be rigid, strong, instead of flaccid. So turgid versus flaccid, and that's obviously due to osmosis. If there was a low concentration out here, loads of dissolved ions, then obviously water's moving out. It's becoming, it's becoming flaccid. But if it was the other way around, then water would move in, and therefore it would become more turgid. So that's another property you could mention, is this idea that it acts in plant cells to provide rigidity. So, and I guess another property of water is literally that it helps with translocation too. So, the xylem. Let's look at the xylem. The xylem is comprised of long tubes of dead cells joined in bundles. So they are joined in bundles. They are comprised of dead cells, which have no organelles, and obviously that's an important property. Similar to red blood cells, how that increases the surface area to volume ratio, they have no organelles, so that water can smoothly move up. So it's for water. So instead of talking about oxygen absorption for hemoglobin, here you're talking about water movement. Their hollow lumen makes an uninterrupted tube. So it's a long continuous tube. That is obviously probably the property that everyone would probably understand. I forgot about this. Lignin. It forms strong walls. And this is it. It is waterproof to prevent water from adhering too much to the surface. So in an exam, I don't know what context you could get asked about this. Maybe a 3-mark question where it's saying something about the xylem. Or maybe you would have to, describe 2 properties of it. I don't know, really. But obviously, there you go. No organelles to help with the smooth movement of water. It's a long continuous tube, and obviously the lignin, which helps with the strength of, oh my god, the strength of the xylem wall, and prevents water adhesion because it's waterproof. See, I forgot about this. I honestly did forget lignin was a thing until just now. If I got into an exam and got asked about the properties of a xylem, I would likely get that, mentioning it's a long tube, but I would probably start yapping about adhesion with no idea of what it is. And there's gaps in the lignin. It allows water to move out. Okay. So, movement of water. This is transpiration. I will have to get rid of this, unfortunately. I'll just use my hand. We'll keep the water though, that's like the central point of all of this. So, the idea is that we have a leaf, and I don't really know how to draw a leaf, because I don't do art. That is a leaf. The idea is that water is going to evaporate off. And you would get a mark literally just for saying this. Now, there is the process written here on the computer. I'm not going to look at that. I'm just doing this off the top of my head now. And we'll kind of see where it goes. So, first step of the process is water is going to evaporate from the leaves. Obviously, the side effect of this is that this is going to lower the water potential. Obviously, WP means water potential. Don't do that in an exam. The amount of times I abbreviate stuff, I nearly start doing it in exams now, which is bad. Like, don't do that, please. Don't abbreviate things like me. That will lower the water potential. Now, I don't know whether it's creating tension or something like that, but I'll just put creates tension. So, water from the roots is going to be pulled up the column due to this tension, because water is cohesive due to, and that's when you would talk about the hydrogen bonds. So, let me have a look. So, yes, this increases tension, which pulls water into the leaf, and that's called transpirational pull. Water molecules are cohesive due to the fact that they form hydrogen bonds. This is known as cohesion. This pulls the whole column of water up from the roots to the tissue, and water enters. So let me look at what I wrote down. I wrote that water in the leaf is evaporated, lowering the water potential. I mentioned that. So that will be 1 mark. This whole section here will be worth 1 mark. This creates tension, so water in the xylem is pulled up the column. I mentioned pulled up a column, creating tension, so that would be, again, another mark. Due to hydrogen bonds being cohesive, that would be another mark. And if this was a 4-mark question, I would have missed a mark, because you would have needed to also add adhesion of H2O molecules to the walls of the xylem. So a model answer would be literally the water would evaporate. I'm just doing it in bullet points. Water would evaporate, I explained that. This would lower the water potential, and that would be 1 mark. Therefore, water is going to move in a column due to the tension. So lower water potential will increase the tension. That's 1 mark. Therefore, water is going to move up in a column due to the cohesion, due to the hydrogen bonds. And then you would also elaborate on the fact that it's because of adhesion of the xylem walls. Not bad. Not bad. Now we move on to the other kind of beast, which is translocation. So transpiration A lot of trans words. I keep getting them mixed up. Transpiration is literally just water evaporates, therefore it moves it up in a column. Translocation, or the mass flow hypothesis, is a bit different. This could be worth 6 marks. Maybe you get an extended response question on this, maybe you don't. Let's have a look. So, I always draw it as a diagram, and I draw it like this. Just to really simplify it for you guys who might not get it. And also myself, because I don't get this, but I will now. Right. Xylem phloem. Companion cells. We have the source. This is our sink. For example, this would be things like, these guys are producing the sugars. What needs sugars? That would be things like growing cells, or, fruits, things like that require sugar, basically, a large quantity. Obviously, they would also be stored as, not cellular, they'd be stored as insoluble starch, or maybe the sugars would be used as cellulose, like I said. So, oh my god, I'm coughing so much, this is annoying. My apologies. Right, step 1. Sugar is going to move out. I'm going to draw it as a hexagon, because we're just saying it's a hexo sugar. Sugar is going to move out, step 1. Step 2, this is going to lower the water potential. Because sugar is soluble. No, starch isn't soluble, but things like sucrose, I believe, are. That is something, digestion. I forgot that was a topic. I need to revise that, definitely. That's going to lower the water potential. Therefore, water is going to move from the xylem into the phloem. This is going to increase the turgor pressure. By the way, that's by osmosis. That will be a mark. That would increase the turgor pressure, establishing a pressure gradient, and forcing the sugar down into the sink, where it would then be used either stored as starch, which, the properties of starch. Isn't that amylose and amylopectin? Or cellulose, which is the long microfibers, unbranched long hydrogen bonds form microfibers. Okay, let's see how much of that we actually got right. So sugars, first mistake, I didn't specify transport. This is active transport. Makes sense, because obviously they're not gonna be small molecules. This lowers the water potential gradients because sugars are soluble, so 1 mark, maybe 1 mark. I'll give myself half a mark for that. That's a mark though. Water from the xylem moves into a phloem via osmosis down the gradient. That will be a mark. Increasing turgor pressure, establishing a pressure gradient. Sugars move into the sink down the pressure gradients where they are used. That could be a 6-mark question. And it is literally active transport, osmosis. That is arguably one of the nicest processes in biology, I would argue. That is such a nice process to really know. And think about it this way. If you get asked a question, importance of water in biology, you could use that, really. You could use translocation, because it involves water. You could say the importance of water is establishing the pressure gradient. so that sugars can reach the sink. Talk about that importance. Oh my god, that is so good. That is perfect. Okay. Let's go look over the structure of... Oh, there is also the extra shit. The cohesion tension theory, which is what translocation is. The evidence for it. Evidence point 1 is during the daytime, where the sun is out, evaporation is high, therefore transpiration is high. Remember, that's the whole gimmick. It's basically just evaporation, but for nerds. So during the daytime, transpiration is at its highest. This is going to decrease the volume of the diameter of things like trees or plants overall, because it's the adhesion is going to cause tension, basically squeezing, pulling in. And during the night, transpiration is at its lowest. So evidence is that during the day, trees or plants are going to be diameter. The diameter is going to be, in the day, smaller, in the night, larger. That's easy. Second bit of evidence, if a trunk is damaged and the xylem is broken, water won't leak out, which implies that they're not under pressure, and air that enters the tree means that the column is broken. So if there was a huge gap here, air is going to enter, so our little column is going to be broken. So that is the evidence to support the cohesion tension theory. So literally, the evidence is if you hug trees, they get bigger, and the evidence is that they don't explode water. I know that sounds stupid, but that is quite literally the evidence. Obviously, you would explain that more biologically. You would say that it's the negative pressure that sucks in or increases the tension. So your evidence there is the diameter decreases in the day because of the increased tension. So that's easy. That is easy. Now let's go on to good old reliable translocation. Then I think there's a practical or two here that we need to do, and then we should be done talking about plants. Oh, no, we're not, because we need to talk about gas exchange and guard cells. So this might be a bit of a lengthier stream, but that's fine by me. It's just my legs hurt when I'm stood up this long, because I'm a lazy fuck. Okay. Right. Comparisons between translocation and transpiration. Obvious differences: translocation happens in the phloem, transpiration happens in the xylem. It uses active transport. It's bidirectional, unlike transpiration, which is unidirectional. And that's it. The idea is, yeah, it's a two-way flow, and they use companion cells. So, they are living cells that basically are called sieve tubes, and they have no nucleus or organelles to maximize the space, just like we said was the case for the xylem. Each sieve tube is supported by a companion cell, which contains a nuclei and the mitochondria for ATP production. That would be a lovely 2-mark exam question, I just realised, where it asks you, describe a property of a companion cells in the phloem. Many mitochondria for active transport, to produce ATP via respiration. Always mention respiration, it's a good point to put. To produce ATP used for active transport of sugars out of the companion cells into the phloem. The end walls between cells, let solutes pass. So they are joined end to end to form sieve tubes. They have lots of holes, and there is a companion tube. Why is sucrose transported? Concentration gradient is maintained by enzymes which break them down. Okay. Evidence to support the mass flow is the idea that sap is released when the sieve tubes are cut, demonstrating that there is a positive pressure, unlike the xylem where nothing flows out. This can be seen in the excretion of sap. The sap is forced... evidence to suggest this is in a xylem, water's moving up, we break the column, then water can't move up. So no water comes spraying out. Meanwhile, with a phloem where we have sugar moving up and down, because it relies on positive pressure, we break it and it's going to leak out. Evidence against it, there is some evidence against it, is that the sugar travels to many different sinks, and therefore it doesn't travel to the one with the highest water potential. I don't think we need to know that, to be honest, so I'm just going to skip that. Investigating transporting plans. This might be a big PowerPoint. Please don't be, please. Oh, it is. Fuck off. Oh, okay, easy. Ringing and tracing. Ringing is where you remove a ring bark, which includes the phloem, but it leaves the xylem intact. This causes bloating above, I believe. Yeah, the stem above the ring swells because there's a huge accumulation of sugar. So yeah, it's this idea, and it supports the mass flow hypothesis. It allows you to investigate it, because what you literally have is we have this is the source, this is the sink. We break this, then it's obviously there's going to be sugars, but they can't go anywhere, so it's going to lead to an increase. Okay. And then tracing is, I think, just using carbon. It's what radioactive? Carbon-14. I love carbon-14 because it's radioactive. Plants can be supplied with it, obviously via photosynthesis. It produces the sugar, and then because the sugar is made with radioactive carbon, you can then trace where it's been translocated at. and you can use X-rays to figure out where the source and the sink are. Cool. Final one is guard cells. Which I'm pretty sure I could do off the top of my head anyways. Let's have a look. So we already know things like the palisade cells are the regular looking plant cells, the ones which contain many chloroplasts. Above them is the upper epidermis. You obviously have the spongy mesophyll layer, which is very airy in order for gas to move in and out. Or loosely packed. Guard cells are obviously the ones controlling what goes in and out of the stomata. So, transpiration is the loss of water from a plant. Stomata can control how much water leaves. Plants in dry environments will have fewer stomata. We know that. Opening cycle. When plants have enough water, guard cells are turgid. When plants are dehydrated, the plant cells become flaccid because water moves out, causing the hole to close. Stomata also close at night when little photosynthesis is happening. So, how do guard cells open and close? What is Luke Kelly saying? Right, fuck off. When light intensity is high, okay, it's potassium ions. When light intensity is high, potassium ions are moved into the cell, increasing the solution's concentration. Therefore, water is going to move moves into the cells, increasing the concentration. The ions make that lowers the water potential, so water moves in, causing swelling. Okay, that's easy. I don't even need to write that on the board, but I will. It is literally that in the guard cells. Potassium ions move in during periods of high light intensity. So during periods of high light intensity, potassium ions will move in. This makes the solution, as expected, more concentrated, which lowers the water potential. Therefore, water is going to move in, which overall, oh my god, makes it more turgid. But due to the thick part, it is basically going to cause, due to this thick part of the guard cell, it is going to cause the stomata to be exposed. And xerophytic cells have thicker waxy cuticles, hairy leaves, sunken stomata, less stomata, rolling up leaves, and a reduced surface area to volume ratio. Okay. I'm going to crack up there because, to be honest, it's getting dark. I'm getting tired. I've done a lot today. But I will happily spend the next half an hour or so writing these down on cards, which is what I've been doing. I have been doing these, not many, bear in mind, but I did a couple of these. I did one on the generic biological molecules just to show kind of a demonstration. But I've done one card on osmoregulation. I've done one card on gas exchange in fish. And one card on gas exchange in insects. Just to roughly explain for each process in and out. So that's what I'll do. And yeah, I think that probably ends it for today's stream, guys. To be honest, obviously tomorrow's history mock day, which isn't going to be favourable at all, but that should be fine. So I'll catch you guys in a bit.